This is part one of the genetic quantitative module. And the first thing we want to deal with is what are quantitative traits. Let's remember that the type of traits that we have dealt with so far are qualitative traits. We can put them into categories such as tall or dwarf. Quantitative traits are different. They vary continuously, which means that we cannot put them in any category, but we have to measure them with a unit. And the unit here, from dwarf to tall, would be, for example, centimeters or inches. These are very common and they are very important. Crop yield, blood pressure, body weight, caffeine content, propensity to disease, age at death, fat content in milk. These are all traits that are useful for human to consider and potentially to address. A disease trait may be the subject of a medical investigation. The content of fat in milk may be subject to breeding. So we'd like to understand how these quantitative traits are controlled and how can we connect genes on chromosomes to these quantitative traits. Before we go any further, let's remind ourselves of what a frequency distribution is. And a good example is the frequency distribution shown here for all the midterm exams from midterm 1 to midterm 4 in our class. And you can see that students are distributed from 400 to as low as 100. And the mean of this distribution is 292. We can calculate how dispersed the distribution is, that is how far it ranges from the mean, by measuring variance. So what is variance? Variance is the summatory of all the values for a given for the distribution minus the mean. So it would be x minus the mean squared and divided by n minus 1, n mean the total number of students in the class. So this is variance. You can also take the square root of variance, and that is the standard deviation. There is another statistical analysis that we can do on distribution, which is to look at two variables and to ask ourselves the questions, are they correlated to each other? So for example, let's look at the data points here for all the midterm exams from, again, 100 to 400. See, for example, here we have maybe this student here has 350 on the midterm exam sum and has about 175 on the clicker total. So we'd like to know, is the clicker total score to February 14 related to the performance in the midterm exams? And one way, of course, is just to look at the distribution here. And the other way is actually to calculate a little more accurately with some quantitative measure. So correlation tells us that the change in one variable is related to change in another variable. Now the correlation coefficient measures the strength of the association from 0 to 1. We can measure that here, and you can know statistically, you can look up the formula for how you measure the correlation coefficient, but I can tell you that in this case the correlation coefficient, indicated as R, is equal to 0.59. So there's a fairly good correlation between your performance in clickers scores and your performance in the midterm exam. We can even get a line that will enable us to predict how a given change in the midterm exam would be matched by a given change in the clicker total. This line is called regression, and it has the very well-known formula of y, that is the performance in clicker, is equal to an intercept a plus a factor multiplied by the performance in the midterm exams. That B factor is the correlation coefficient. What it is the amount of change in y on a given unit of x. Now, getting back then to inheritance of traits, it was obvious very early on in the analysis of genetics, that is in the early 1900s, that many traits were polygenic. What does it mean? It means that they corresponded or they derived from the genotype of multiple loci. And one of the early examples demonstrated for this is the case of kernel color in wheat, and in fact ear color. So there are some varieties of wheat that have the ear, that is the structure that holds all the seed, and it has these little awns or these little hair sticking out. That can be either dark purple or it can be white. If you take inbred parents that breed pure for these two characteristics, and you now cross these two and derive an F1, you find that the F1 is intermediate. It has a color which is halfway between the one of the two parents, is the red. If we then take the F1, self it, and derive F2s, the F2s fit ideally into 5 classes. 2 classes that are the parental, pure white and purple, and then 3 intermediate classes, which we'll call dark red, red, and light red. It was demonstrated that this is due to the genotype at 2 different genes, one being A and the other one being B. And I'm going to represent them very simple like this and show you that the genotype of the dark purple parent was in fact big A, big A, big B, big B, while the genotype of the white parent was little a, little a, little b, little b. Of course, we know the heterozygous genotype. And now we should be able to calculate what is the genotypes for the different classes. Now, we need to understand, though, what is the rule? How does this polygenic inheritance work? You can think of it in a relatively simple way. If you have an uppercase allele, so let's call it X. X gives us 1 unit of color. And if you have little x, we get 0. So if you think of it that way, you can then see how having 4 uppercase alleles gives us the deepest color. This is the 4 alleles. And then having 0 of these uppercase alleles gives us the lightest color, which is white. And then everything in the middle would be intermediate. For example, here's a class that has 1 color allele, and it could have just a big A or just a big B, and it would give us the phenotype light red. Here's another class which would have 2 alleles being uppercase, such as big A, and then being little b, or perhaps being big B, and then big A, little a, and that gives us a red color. And finally, we get a class which is dark red, which has 3 color alleles, and again, this would be, for example, big A, big B, and little b. Now, you wonder about this and say, can I figure out what the frequencies of these different classes is? And the answer is, well, it looks perhaps a little bit complicated, but it's doable. Now, before I tell you about that, let me show you the concept of additive inheritance. We just stated it. And it was as follows. Each uppercase allele adds 1 unit of color. Each lowercase allele adds nothing. So when I look at that, at any genotype, I can then calculate in an additive fashion what the phenotypic output should be. So for example, let's think of height. And let's consider first on this side a case of additive inheritance. And so on the left here we have one parent, let's say big T, and he's got the largest size. And over here is the other parent, little t, and he has the smallest height. And in the middle is the heterozygous. It could be, for example, the F1. Now, if we look at what happens with big T, we can actually think of that phenotype being the results of the two T's, each contributing a given portion of height. That is, functioning in an additive sense. And over here, for the little t, homozygous, we could think again of the total phenotype for that trait to be the result of the additive action of the two little t's. So let's look at that a little closer, like this. Then, if I look at the phenotype of the heterozygote, I can actually predict it. And I see now that I've not drawn it entirely right, so let's say that this thing was here and then here, by having one big T and then one little t there. So this is a very nice situation from a geneticist point of view, because it means that if you know the genotype, you can predict exactly or fairly accurately what the phenotype should be. Look instead at a situation for a dominant trait. This is a dominant trait, and here we don't have additivity. What we have is big T equal to big T, little t, and little t, little t. Now we understand it, but there is a class here that we cannot predict genotypically, meaning that any one of these individuals that has that height could either be homozygous for big T or heterozygous. How do we calculate the frequency of phenotypic classes? And by the way, we're going back to our color alleles. So we have the 5 different classes: purple, dark red, red, light red, and white. And we have 2 loci that are determining the phenotype for this trait. So the assumption is that all loci are independent. They're all equivalent in contribution to, to phenotype. And that there is therefore a probability connected to having an upper letter allele, which you're going to call P. Here it is. And then there is the probability of getting the lower letter allele. We're going to call that Q. And of course, in this case, the 2 probabilities are the same. So P and Q are both 1/2. And then there is a way that you can think of this problem as the total phenotype resulting from a genotype that has been formed in this way. We essentially have 4 equivalent slots. Now, of course, one is for the A gene and one is for the B gene, but that doesn't really matter, and we can consider them almost as similar or functioning the same way because each one contributes the same unit of color. And then we either put, in each one, we either put a large letter or a little letter allele. So it's the same for this one and for this one, and so on and so forth. So they each can get one or the other allele in that position. I can then calculate what is the probability for one of these arrangement. Now you recognize by now that probably that this arrangement is starting to look very similar to the one that we treated with the binomial expansion. So for example, let's ask ourselves, what is the probability of getting genotypes that correspond to one large allele, such as big X, and 3 small alleles? To visualize this, we can just proceed here, and I'm going to fill it as big X, as little x, and little x. Then I can put a little x in this one, and then a big X in this one, little x, little x. Let me make sure this one looks like a big X. There we go, big X. And now over here, I could actually have it in the 3rd slot. So as a big X in the 3rd slot, and a little x in the fourth. And then over here, I could have a big X in the last one. So, of course, this could have been A and B, but the fact is it doesn't really matter. There are 4 possible ways in which you can form this type of genotype regardless of the position of the large X. Now, for that, we actually know how to deal with. And the answer is that to deal with that, we can take the Pascal triangle. And this type of situation is the one that can be identified by this row. So this is the row for 4 slots. This is the row where we can determine the number of combinations for the 2 potential outcomes at each different slot. So if we look then at this number, let's go with the red here. If we look at this number, it means that there is only one way of getting, for example, little x, little x, little x, and little x. Now we just measure here the second case, we just determined that. There are 4 ways of getting this type of genotype. Now this one says that there are 6 ways of getting the genotype in which, for example, we have 2 little x and 2 large x. And then 4 ways again of getting the genotype where we have 3 large x's and 1 little x. And finally, there is 1 way for us to get a genotype with 4 large allele letters. So these are the coefficient of the binomial expansion. The probability for any given genotype can be calculated. We know that, for example, in this particular case, let's do the calculation in blue. We have 1/2 times 1/2 times 1/2 times 1/2. So this gives us 1 out of 16. So the probability of any one of the possible genotypes is 1/16. These are the coefficients. So for the situation shown here, we can take the 6 and multiply times this one. So we have 6 combination out of 16 that would give us the outcome that we wish. So now we can go to our categories over here and fill in the frequencies. So the one with all large case alleles is 1 out of 16. The one with 3 is 4 out of 16. The one with the one with 2 is 6 out of 16. 4. And then 1 out of 16. So we have determined all the possible frequencies. Now this is, how would you do if you have to go for 3 loci? You have a pod with 6 different slots. So we would have to take this one. And for example, if we wanted to calculate it for 3 loci, what is the probability of getting a genotype which has 3 large case alleles and 3 small case alleles, such as, for example, big A, little a, big B, big B, and then little c, little c. That's one of them. Well, but to get them all, we would have to do 20 times 1/2 times 1/2 times 1/2 times 1/2 times 1/2. So we have 1 out of 64. So our frequency for this case would then be 20 out of 64. That is for all the possibility of having 3 alleles of one type, the large type, and 3 alleles of the small case type. There is a funny thing to notice about the Pascal triangle, which is that if you take the sum of all these guys, you get 16 in this case, which is exactly your denominator. And over here, if you get the sum of all these guys, you get 64, which is again the denominator for the frequency distribution. So that's a pretty interesting way of using the Pascal triangle, isn't it? But otherwise, you can calculate it simply by multiplying the probabilities for each little slot. So essentially what we've learned is how to calculate the frequency of each phenotypic class. Now let's go on and ask ourselves what type of phenotypic variation we would expect. So if I look at the population here in the F2, what do I actually expect? Something like red color is not going to be perfectly red. It doesn't have one exact specific color, but there's going to be other factors that somehow affect it, mostly environmental factors. Perhaps the soil, the type of weather, amount of water, and every plant is exposed to somewhat different environment. Therefore, when I look at the classes that I would be getting, I might expect to get very sharp divisions of these classes. So I have these 5 classes divided in this way, right? Where here is the fully purple, here's the fully white, and then in the middle, for example, is the frequency of the fully red. But in reality, you know that this distribution is not going to look like that. The way it's going to look, it is going to look something like this, and depending on how much of an environmental effect there is, it may even look like this. Looking at distribution, you may not be able to tell the number of classes. Because the class boundaries are blurred, you want to determine whether there is in fact an environmental effect. Now, how would you tell that? Well, the way to tell that would be to look at the parents. If you look at the parents, you would be expecting exactly this type of distribution, right? The parents would look, actually, if you just look at parents, let's look at it this way. But in reality, even the parents would vary, perhaps. You might have instead a distribution that looks like this for the parents. And then the same thing could be said for the F1. So the F1, again, you expect the F1 to look like this, but instead you may get an F1 that looks like this. So this would tell you that this deviation from the ideal center here is the effect of the environment. And then the total phenotypic variation is actually a result of both the genotype and the environment. How do we then factor the ability of assessing these multigenic traits and at the same time breeding them to obtain progeny that have a given genotype? Such a case was addressed by Edward East as early as 1916, when he was studying color, flower length in Nicotiana. And what he observed was that he had 2 pure breeding parental strains, and there was no doubt an environmental variation then in this in both of these strains, because they would distribute around a mean, and the mean was different. In fact, you can see that these two don't overlap. Here the largest corolla is about 45, and here the smallest corolla length about 87. Now if you then go to the F1 generation, you find that a similar type of variation. Now let's be scientific, let's call this variance. The variance observed here was in fact comparable to the variance that has been that was observed in the parents. And then, or when East got to the F2 generation, he saw now that the variance was much larger. So again, variance is a measure of the deviation from the mean, and remember it is a square. It would be a number. We can actually calculate it. Let's say that S is equal 5. It's about something like this. Then the variance would be 25, which is the square of the standard deviation. Anyway, so the variance here would be much larger than the variance here, and so you knew that you had a genetic contribution here, and the genetic contribution would be the difference between this variance and that variance, or H2. Does that make sense? So let's break down the possible sources of variance. So the total variance in a population is going to be called the phenotypic variance, VP. And VP, we can determine it has a genetic component. We're going to call that variance G for genetic or genotype. Plus it has a variance due to the environment, so that's going to be called VE. And then it has a variance due to the interaction of the genotype with the environment. Now this variance may be a little mysterious to you. It's very interesting, but I won't talk about this. So for the purpose of this lecture, we can effectively forget that, although don't forget it entirely, it does exist. The heritability, the amount to which a phenotype depends on the genotype, can be derived very simply by dividing the variation due to the genotype over the total amount of variation, which is VP. So this is called heritability, and because heritability is still a variance, we have it squared. So large H squared is heritability. Let's think though about the types of genetic component that give us the variance. That is, if I look at a trait and I'm asking, why does the trait vary? It could be because the genes involved, or a gene contributing to this trait, is exhibiting simply dominance, right? So, for example, a trait may follow the type of distribution that I showed you, where big T would have this height, then big T, little t would have this height, and little t would have this height. Now this, we call it additive, right? But remember, there was another one which we're quite familiar, which is the dominant manner of inheritance, where the big T, little t is identical in phenotype to the big T homozygous. So this little t. Now this is dominance. So dominance is actually a bit tricky to us. Why is it tricky? Because when I look at this type of plant, when I look at a plant or at an individual or an animal or a man or a woman with this type of variation, I do not know whether I'm looking at the heterozygote or at the homozygote. So there is a variance which is due to dominance that is certainly part of my total variance. So I can actually start writing that out and say the genetic variance Vg is equal to the variance due to dominance Vd. Then there is another source of variance, which is epistasis, or interaction between loci. You're familiar again, and without going into the details of it, you remember that epistasis can completely hide a genotype under another genotype. So that in fact is even more tricky than dominance, because with epistasis everything could happen. The type of variation that is due to gene interaction, you may remember on the Punnett square, it meant that if we had recessive dominance, a certain compartment could not be even scrutinized. And if you had dominant epistasis, it would be even the larger part of the population would be subjected to this effect. So clearly, it's a tricky, non-desirable component when we really aim to determine rapidly what we have. So that component exists, and so we call that VI. And then the last component is the component that we can really control, because it's very easy. Every phenotype corresponds to a specific genotype, as that the additive component. Of this total variance, this one, I'm going to call it not so easy or hard to model. And this one is easy for me to follow. If I'm really interested in knowing what heritability I have, I might just want to disregard the additive and dominant component of my variance and just look purely at the additive one. And this is reflected in the fact that, in fact, there are two types of heritability. One is the broad sense heritability, which is this one, which is the total variance due to genotype over the total variance due to phenotype. But over here, there's the narrow sense heritability, which is in fact a better, easier heritability for us to consider, which is the total variance due to additive effect, additive genetic effect, over the total phenotypic variance. Narrow sense heritability is what a breeder wants to know, because it predicts the success of a selective breeding. So how do we estimate broad sense heritability? So the total heritability, or the broad sense heritability, can be calculated by looking at the type of variation that we have, or variance that we have, in populations that have individuals of identical genotype. Let's look at this graph. This is the graph of, or the distribution of corolla length for parental strain A and for parental strain B. The variance that we see here cannot be due to genotype. They all have the same genotype. Therefore, the variance here is due to the environmental effect on the development of this corolla length. And the same is due here. So these two variances are environmental variances. And in fact, we see that they are about the same. Not only, but if we look at the F1 generation, this one also has a similar variance, which is also due to the environment. Now, what happens though when we look at the F2? Well, in the F2, we are going to see two different variances at work. One is the variance due to the environment, and the other one is the variance due to the genotype. Now, how do we estimate the variance due to the genotype? Well, if you just look up here, the only variance at work here is the variance due to the environment. So it means that in this individual, the variance, the total variance, was the variance due to the environment. And in this individual here, the total variance, VP, is due to VE plus VG. So then how do we calculate VG? Well, it's pretty simple. We then take the total phenotypic variance, we subtract the environmental variance that we have determined using these individuals, and now we can use this genotypic variance to derive the heritability. The total genotypic variance over the total variance is H2, or H squared. Simple enough, right? Now, how do we derive narrow sense heritability? Well, for narrow sense heritability, we determine the correlation between offspring and parental phenotype. There are actually multiple methods, but the one that really makes a lot of sense to us is to look at this. If we plot the relationship between the parental phenotype on the x-axis versus the offspring phenotype, we might have a situation such as this one, where, in fact, there is no correlation. You can tell that these points are all over. There is no way that we can predict one from the other. We cannot predict offspring phenotype from the parental phenotype. On the other hand, here, we have a very nice correlation, and the slope of this correlation is, in fact, the narrow sense heritability. The regression coefficient B, remember, we're looking at an equation where y is equal a plus bx. So B in this case is nothing but our narrow sense heritability. And in fact, it shows here that for every unit of increase in parental phenotype, we can expect a unit of increase in progeny phenotype. So there is a perfect correlation of the genes you get from your parents to the phenotype that you manifest. Now here you see that the correlation is not as perfect. I mean, they're highly correlated, but the slope is not as distinctly covariant, and therefore our heritability is only half. Why does this matter? It is very interesting to determine heritability. Now you may wonder, oh yes, I like to know heritability because maybe I want to know whether IQ is heritable. I want to know whether my good look is going to be heritable to my children. Now, there are many misconceptions to the concept of heritability. Now, heritability, all it is really the result of a formula that is dependent on the environmental variability and the genetic variability. And it applies to a population. You cannot calculate variability for anything but a population. Here's a number of false interpretation or misconception. For example, we may think that heritability tells us the extent to which a trait is genetic. This is false. It is dependent on this for a given population, and both terms vary greatly. So they may vary from one population to the other, one environment to the other one, so that there's no real certainty that a trait that has zero heritability in one population will also have zero heritability in another population. It doesn't tell us anything about what component of an individual phenotype is due to genetic inheritance. That is, an individual does not have heritability, a population does. Heritability is fixed for a trait. Again, misconception. It changes depending on population and environment. This is almost a repetition of this one. And then here, population with different heritability for a trait are genetically different. Again, this is false. It is not so. The environment could be different. They could have hugely different heritability, but just be in a different environment. And then traits shared by a family have high heritability. That's not so. Family members share environment, and therefore they would be equally affected. So keep all this in mind, and when you have time, look at the second part of our quantitative genetic lecture.